Good morning, everyone. Today I would like to talk about this paper called Single-Cell Derived Tumor Sphere Formation and Drug Resistance Assay Using an Integrated Microfluidics. So it's important to study cancer stem cells as first because they promote metastasis and they enable drug resistance. That's why it's very exciting to look at the single cell derived spheres from glioblastoma. And what they did is to test two different cell lines, the standard glioblastoma cells and an induced cell line, which is going to be smaller and deformable. That's why it's very interesting that they demonstrated that the biomechanics of these tumor cells affects the formation of the spheres. So what they did, here you see that they depict the prominent or less deformable cell in blue, and the small or more deformable cell, the induced cell line, in purple. So here is the mixture of these two cell lines, and they are going to be injected in this microfluidic device. Here you see the direction of the flow. And this is a microfluidic chip that has many matrices. So here is the single cell capture procedure. So here you see that the pores are bigger than in the next stage. And after culturing the cells, and once they are located in these chambers, they are going to invert the chip. And that's how these cells enter to the culture chamber. After the sphere formation, they are going to introduce the reagents. That is the vincristine drug. That's the one that they tested. So here we see a zoom in of these matrices. So here you see the dimensions, and it's important to notice that the pores, this pore called R, is 2 microns bigger than the next pore called lower R. Here you see the dimensions, and this device is called multi-obstacle architecture looking. So this is why it's called MOLE. And they try different flow rates to obtain the optimal conditions. And they found that at 100 microliters per minute, they could improve the capture of the cells. So here we see the flow direction and the fluorescence. The cells are marked with fluorescence to evaluate if there is just one cell per chamber. And that's what you see here. In the merge image, you see the chambers and just one cell located in them. So the distribution of the cells, here you see in blue the standard cell line, and in red the induced cell line. You see how they distribute in these different sizes of the microunit matrix. So when the pores are 14, 12, 10, 8, and 6. And they also count the number of cells that they have in each of these places. And this is a very nice image of how the spheres, first the cells at day 0, we have the cells trapped here in these chambers, and then as time goes by, for example, in day 5, the spheres they have formed, and at day 10, we see that the spheres have grown. And in A, we have the standard cell line, and in B, we have the induced glioblastoma cell line. They compare the sphere formation rate of these both cell lines and also the size of the sphere in square micrometers. You see that the induced cell line has grown more. And they also checked the cell viability. They did it by measuring the mitochondrial potential, and also when they use this drug vincristine, which is a microtube targeting agent, and it irreversibly binds to the microtubes and spindle proteins in the mitotic S phase, how it produces apoptosis. So they also measure the caspase. Here you see, they compare the standard cell line and the induced one, and the induced one has less damages. The percentage of caspase 3 is lower, and also the mitochondria has less damages. And that's how they demonstrate that the induced cell line, the smaller or more deformable, could have more resistance to vincristine than the standard, more prominent or lesser deformable cancer stem cell derived spheres. All right, and just to finish, I wanted to show you the supplementary information, which was very interesting. This graphic, the figure S4. Here it is. And here you see a lateral view of the cell culture chamber layer, which is depicted in gray. And in the first stage, when they introduce the cells, here is where cells are going to locate. And after the cell infusion and the single cell capture, they are going to perform the chip inverting. So they invert the chip, and that's how the cells are going to be now in this culture chamber. And that's how the spheres are going to grow in this culture chamber, which is wider and more comfortable for the cells to grow. Then the reagents are going to pass. And they have also performed flow simulation, and they demonstrate that the flow is very comfortable for the spheres to grow over there. All right, thank you very much.